Okay. Well, thank you. Thank you for inviting me. Even though I'm not too far away, it's actually my first time on Yale campus, so very excited to be here. Chi, thank you for the invitation. So I'm going to be talking about, So I'm going to be talking about some AI-enabled techniques for PET image enhancement. Okay. All right, so the quantitative accuracy of PET is greatly diminished by high noise and low spatial resolution. On the noise front, of course, there's the scan duration and radio tracer dose. These are all operational factors. On the resolution front, there are hardware aspects and software aspects that determine what spatial resolution we're working with. So my lab has been working on a bunch of different denoising and deblurring techniques, all image domain, going from corrupt inputs to clean outputs for PET images. All right, so this is my summary slide, putting together a lot of developments that happened over many years. So there were lots of omissions, please pardon that. But yeah, for the most part, the trend that we have observed is initially in the early years, we saw a lot of shallow CNNs to deeper CNNs. And then at some point, we started seeing UNets being used for more than segmentation. And then it kind of completely took over the image generation, sort of became the default network to start off with for image generation. And then I do want to mention, like, I feel like with this GAN, from going from the GAN to the cycle GAN, we really, this change really facilitated this parallel transition, which is the reliance on fully supervised architectures to more unsupervised and weakly supervised architectures. And then, of course, since then, we have seen a lot of very interesting methods come up on the unsupervised side, including the deep image prior and diffusion models, which we've been extensively discussing at this meeting. So one of the things we've been interested in is SR PET imaging. And so I'm going to be talking about both of these paradigms initially, where we were looking at paired low-res, high-res images. And then at some point, we switched over and decided to look at unpaired low-res and high-res images, where we've got two cohorts, one at a certain resolution level, another at a different one, and these are not from the same patients. So for the most part, we've looked at looking at this mapping from the HR plus scanner domain to the HRRT scanner domain. The HR plus is a legacy clinical PET system. We have quite a bit of data available across many different cohorts and disease types for the HR plus, which is why we thought this was a good starting target. And the HRRT is a dedicated human PET scanner, dedicated human brain PET scanner. All right, so this was some early work we did on the SR PET. So again, this is supervised learning, and we tried to incorporate high resolution anatomical information to guide the process. We use residual learning. And these are some simulation results, which were based on the BrainWeb Phantom. And this was trained on a perfect ground truth. And we looked at a lot of different comparisons at that point, how very deep networks are different from shallow networks, how different combinations of inputs, including spatial inputs, which are basically looking at the radial and axial coordinates of each pixel in the input image, given the fact that spatial resolution for PET is spatially variable. And so putting all of that together, we of course got the best results, but this was for perfect ground truth. And so we moved on. Even in our simulation studies, we started looking at an HRRT ground truth version, which takes us a little bit closer to clinical reality. And so what did hold up is the general trends in terms of how these very deep versus the shallow versus penalized deconvolution type approaches compared against each other with very subtle differences. And then we extended this to clinical data using an HRRT ground truth, and we got more or less consistent results between simulations and experiments. So the next step of this work took us to self-supervised super resolution, where, so there we are basically trying to work with two different cohorts, low-res, high-res, unpaired. And so this is a very cycle GAN-like architecture. We have got two sets of generators and discriminators, one performing low-res to high-res mapping and another performing the reverse mapping, and there's cycle consistency imposed. One important thing here was this piece. As you can see here, we've got a split generator. What this really does is provide simulation guidance. So we have got this pre-trained VDSR CNN based on simulation data, and we actually split our inputs to create one direct set of inputs and one set of features that are derived from simulation guidance. And with this, we directly jumped into clinical data and we got very interesting results. And we actually kind of went through it part by part, looking at without simulation guidance versus with simulation guidance. And we also used this alternative approach where we did the high res to low res transformation using an analytical deblurring, spatially variant deblurring framework. So we generally got interesting results where some of these subtle features that were only there in the high res image were reliably extracted in this, it's the final version with the simulation guidance. All right. Okay, so I'm gonna move on now to my denoising piece. So again, with denoising, the part that I wanna focus on are cases where your only inputs are corrupt inputs. And so some of the early examples that we've seen in existing literature involve multiple noise realizations. So in this example, we've got 2 noise realizations, but both of them are corrupt. You still want to generate a clean image from there. And then some of our newer work pertains to this other paradigm where you just have a single noisy image and you're trying to get clean images from there. And so in terms of multiple noisy inputs, the popular model for a long time was noise to noise. It's a weakly supervised approach. But again, the limitation is that you need at least 2 noisy inputs. And then there were different variants of this came up of note as noisier to noise. The reason it works with one single noisy input is the fact that it assumes a statistical model. And that's often quite a challenge for an imaging modality like PET, because as Q mentioned in his presentation, there are a lot of these uncertainty estimation approaches, which by themselves are very challenging. So it's not very trivial to incorporate a very dependable noise model. And so we looked into this noise to void approach, which again, it relies on single noisy images for training. At its heart lies this blind spot idea, where we have a masked receptive field, which looks like this. Now, if you have no masking, which is sort of the default when you're implementing convolutional layers, what would happen is your input and your prediction, just because there is just ones here, it's an identity operator, you're gonna get identical input and output here. But on the other hand, when you apply this masking scheme, you're really trying to make your best guess of the central pixel from all of these neighboring pixels. And that's really what the estimation problem is trying to do. And as a result, you get an output which is different from the input, and the central pixel is often nicely estimated. So for its implementation, we use this UNet architecture. It's downsampled as the filter numbers are increasing and so forth. And then there is upsampling, so standard stuff. One thing that made a huge difference was the idea of population level pre-training. And this is a concept that kind of goes back into sort of machine learning theory in terms of there being a lot of evidence that pre-training effectively acts like a good regularizer in these scenarios. And so the motivation, of course, is clear when even though these methods are unsupervised, when we're training case by case, it's a lot of training time. We have to save these individual models for each patient and so forth. And so the idea here is to have a transfer learning paradigm where we're instead of training each model from scratch, we are performing group level pre-training. So, however, this is all using noisy data. So this is all a bunch of noisy images with no ground truth. And we are pre-training our model, and this is an imperfect model, and then we're fine-tuning for each of these individual cases. And so it really helped improve the model's image quality and network overall the stability of the network in the training phase. And so I'll show some results. I don't know how I'm doing my time, but these are simulation results. We looked at different count levels, and I think what would be, and okay, so in terms of the trends, we looked at both more traditional denoising approaches as well as different noise to void variants, including MR guided, without MR, pre-trained, not pre-trained, etc. And so we saw a general benefit of incorporating anatomical information and incorporating the pre-training step. The other thing we noticed was in general was that with pre-training, when we look at these different images, we see some reduction in bias, especially in these regions. Note that this is noise to noise. My personal feeling about noise to noise is that even though you have those two noisy inputs, you do see a lot of sort of over smoothing in different regions. And so it's kind of like a lasting issue. And that's one thing that we are trying to address in some of our more recent work, and I'll cover it very briefly. But okay, so with this, the pre-trained model gave the most promising results, and these also extended and actually were more prominent at the, sorry, I believe I did not update this image, but at the low count level, where we actually saw a much stronger denoising effect with the pre-training and with the incorporation of anatomical information. And these are some clinical results with the same model. There are a lot of these noisy features, which are definitely much more pronounced in some of these other methods that we compared it against, and the pre-training as well with the MR information removes it more effectively. So the other method that I was going to talk about was neighbor to neighbor. So this is also, you can consider it a noise to noise variant. It's the basic concept, as I'll try to explain, involves subsampling a single noisy image to create two noisy versions of the same image, and then apply the noise to noise denoising concept to it. There's also a little bit of an addition here in terms of there being a regularization step where we are really trying to enforce a regularization penalty in the subsample domain, because by default these two subsampled images are different from each other. However, in an extreme theoretical case, these are supposed to be identical to each other, and so that's what enforces. That's what causes this term to be different from that term. And so these are some early preliminary simulation and clinical results from this work. The abstract is still under review, and one notable thing was in terms of PSNR and SSIM, this method actually outperformed the noise to void approach that we have extensively studied. So that's all I've got in terms of our research updates. I do want to make a couple of comments that with all these methods, it is very important to sort of keep in mind that some important concepts of replicability, reproducibility, and generalizability. And so as part of the SNMMI AI task force, we've been looking into some of these aspects very closely, and we have several of these guideline papers. There are also a lot of different methods that are beyond the scope of this short talk that we reviewed recently in this paper, which was a collaboration with Chi Lu. And I just wanted to thank my wonderful, wonderful research team and my funding sources. Questions, please.
Just a detailed question about the neighbor to neighbor. Does that mean you also apply a spatial offset between the two?
That's a good question. So basically what we try to do is we sort of randomly sample every little square box with 4 pixels, and we assign 1 pixel, 2 disjoint pixels to each of those subsampled images.
So it would generate a little bit of spatial blur in both images then, maybe?
Well, that's actually a very interesting question. So there is a little bit of a spatial blur that would result. However, again, the denoise image is in the regular grid size. One of the things that helps is in the regularizer that we implement, we actually have a tuning parameter. And this is the part where this method is actually in a lot of ways an advance over noise to noise, where you can actually tune the level of regularization by varying the weights in terms of how much of a difference between those 2 patches do you want to incorporate, because they're fundamentally different. There could be different due to noise, but they could also be different due to the actual signal content between those 2 patches being non-identical. So I see couple of questions here. Both of them relate to figures of merit, so I'm going to answer them together. And so these actually are very important questions in terms of, like, for the most part, we've used SM and PSNR for evaluating our approaches. And it is a fact that they don't cover the full span of evaluation techniques. So I think what I personally believe in is we have to approach it case by case, because eventually what matters in any medical setup is task-based evaluation. So these two kind of should go hand in hand, where you have image domain evaluation metrics that really talk about your quantitation, and then you have task-specific metrics, which can give you case by case sort of more clinically meaningful results.
Joyita, very interesting work, particularly the labor to labor. We actually implement like about 10 different methods for denoising for lateral imaging, not for PET. And labor to labor actually get the best result way better than other methods. I'm just curious, I cannot stop thinking that since you present super resolution, then labor to labor, you can combine these two together. One is that you decompose the image to two images, right? With lower resolution, then you do noise to noise, and you put them together, right? And PET images already quite a low noise and no resolution. Can you do super resolution and do labor to labor?
So I think that's a good question. So my thoughts on that are actually what I've been thinking on that. It's sort of a thought process, was actually using that as a way to incorporate anatomical information, because typically from an anatomical image, you can actually get much more detail. It wouldn't just be taking the PET image and downsampling it and getting two subsampled images that are coarser and have worse resolution in themselves. And so the original idea that I thought of was kind of doing this in the MR domain, where these MR inputs are split, and basically the expectation there is you can actually enhance the resolution in a much more significant way. All right, thank you, everyone.